Okay, so let's see structure of a nucleotide. Nucleotide. You know, nucleotides are monomer units of DNA and RNA. And in DNA or RNA, nucleotides are joined together. Nucleotides are joined together by condensation. और इनमें जो bonds बनते हैं, they are known as phosphodiester linkages, and we'll discuss how these bonds are formed. So let's see structure of a nucleotide. A nucleotide has three components. Number one, a pentose sugar. There are two types of pentose sugar. There is ribose sugar and deoxyribose sugar. Ribose sugar is found in RNA. It is found in RNA. And deoxyribose sugar is found in DNA. RNA stands for ribonucleic acid. Nucleic acid. And DNA stands for deoxyribonucleic acid. Okay. अच्छा जी. So this is the pentose sugar, and the second component is a phosphate group, and third is a nitrogen base. So there are two types of nitrogen bases. एक को हम कहते हैं purine bases, और एक को हम कहते हैं pyrimidine bases. What is difference in purine bases and pyrimidine bases? Purine bases have a double ring structure. One is a hexagonal ring and other is a pentagonal ring. Okay. So this is purine. And pyrimidines are single ring structure and they are hexagonal rings only. So this is single ring structure and this is double ring structure. So purine bases are larger than the pyrimidine bases and they are heavier. And there are two examples of purine bases. One is represented with an A and is known as an adenine. And other is G and it is guanine. इसी तरह से pyrimidine bases जो हैं वो तीन तरह की हैं. It is represented with C, which is cytosine, and it is P, which is thymine, and it is U, which is uracil. So any one of these five will be present in a nucleotide. And out of these two sugars, any one of them will be found either ribose in RNA or deoxyribose in DNA. So basically there are two basic types of nucleotides. Nucleotides की दो types होंगी. There are RNA nucleotides and there are DNA nucleotides. RNA nucleotides have ribose sugar. Okay, so they have this sugar which is ribose, and they have a phosphate group attached with it, and they can have a nitrogen base here, any one of the 4 nitrogen bases that are found in RNA. Similarly, DNA has deoxyribose, and it has a phosphate group attached with it, and it also has, a nitrogen base here. So on the basis of the different, this structure is the same, in all RNA nucleotides because they have a ribose and phosphate group, and this is same in DNA nucleotides, but they differ from each other. So there are 4 types of RNA nucleotides, 4 RNA nucleotides. They will have ribose and a phosphate group attached with them, and here they may have an adenine. Then they will have ribose here, they will have a phosphate group here, and they can have guanine here. Similarly, they will have ribose here and a phosphate similarly, and they can have cytosine here. जी अस्सलाम वालेकुम जी वालेकुम
सर जी आप ऑनलाइन क्लास ले रहे हैं?
जी मैं ले रहा हूँ.
अच्छा चलें ठीक है. वो हमें साजिश का नजर नहीं आ रहा था इसलिए पूछ रही हूँ.
अच्छा वो लिंक मैं क्लास में शेयर किया हुआ है, वो आपके साथ शेयर कर दूं.
क्लास में किया अपने A1 ग्रुप में?
हाँ A1 में ग्रुप में शेयर किया हुआ है, वहां से ले ले.
चलें मैं चेक कर लेती हूँ.
ठीक, ओके. अच्छा ये स्कूल की जो जी है ना आपकी ईमेल, उसके थ्रू नहीं आप ज्वाइन कर पाएंगे. उसके लिए आपको जो आपकी पर्सनल जीमेल है उससे आप ज्वाइन करें. अच्छा जी. So you have this ribose. So there is another RNA nucleotide which is a phosphate group here, and it has uracil here, okay, instead of thymine. There is no thymine in RNA nucleotide. So these are 4 RNA nucleotides. Similarly, there are 4 nucleotides found in DNA, but they have deoxyribose sugar, a phosphate group here, they have adenine here, they have phosphate group here, they have guanine here, they will have phosphate group, and they will have cytosine here. DNA nucleotides differ from RNA nucleotides in 2 ways, and they would have thymine here and phosphate here. So this is deoxyribose sugar, this is deoxyribose sugar and deoxyribose sugar. So can you tell me what are the 2 differences in between this RNA nucleotides and DNA nucleotides? These are DNA nucleotides, and these are RNA nucleotides. Can you find 2 differences? Are you people there?
The main difference is the sugars.
Yeah, there is a difference.
इसमें डाईऑक्सीराइबोस है.
हाँ, एक तो शुगर का डिफरेंस है. RNA nucleotides have ribose sugar and DNA has a deoxyribose sugar, but the other difference is this. You know, RNA nucleotides will have uracil, RNA will have uracil, and DNA will have thymine instead of uracil. So these are two differences between DNA and RNA. Okay.
Okay, sir.
Now let's see structure of an ATP molecule. Structure of ATP. What is the structure of an ATP? ATP stands for adenosine triphosphate. This is an RNA nucleotide. RNA nucleotide, and this is double phosphorylated nucleotide. So as it is RNA nucleotide, so it will have a ribose sugar. This is a
सर आप एक सेकेंड नीचे स्क्रॉल कर सकते हैं?
जी.
से जीरो लिया उसने वजह
एक सेकेंड.
Double phosphorylated nucleotide.
ठीक है
ठीक है सर, थैंक यू.
Okay, so what does it mean? This is ribose sugar. It has a nitrogen base attached here, which is adenine. We can represent it with A, and a sugar which has a nitrogen base attached with it is known as nucleoside. And because this is adenine attached with it, so it may be known, it is known as adenosine. Okay, so when there is one phosphate molecule attached with it, phosphate group attached with it, then it will be known as a nucleotide. And this is known as an AMP, which stands for adenosine monophosphate. Okay, so this is one nucleotide. If there are two more phosphate attached with it, okay, so if there are these two phosphate attached with it, this would be still a nucleotide, but this would be known as an ATP, which stands for adenosine diphosphate. And you know, if this is the third one, so this would be known as an ATP, which is adenosine triphosphate. Hope you have understood it. So you see, this is a nucleotide, and there are two additional phosphate groups attached with it. So this is why it is called double phosphorylated nucleotide. इसलिए हम इसे double phosphorylated nucleotide कहते हैं. Is it okay?
Yes, sir.
Great. Now let's see. We have this describe the structure of nucleotides, including the phosphorylated nucleotide ATP. Structure formulas are not expected, so we don't need to know the structure of ribose sugar or deoxyribose sugar or nitrogen bases, etc. So figure 5.2 is an incomplete diagram of ATP molecule. This is sugar, this is base, and this is phosphate group. So sugar is a ribose sugar in this case. And what is the nitrogen base? It is adenine. So this is actually an adenosine triphosphate. Okay, but this is one phosphate, so this would be an AMP, adenosine monophosphate, AMP, as there is one phosphate G. Okay, so complete the diagram of the ATP molecule. In an ATP, there should be 3 phosphates: adenosine triphosphate. So one phosphate is already there, so we have to make 2 more phosphate groups to complete this diagram. So now this is an ATP. Write the name of the bases, base in the spaces provided, and write the name of the sugar in the space provided. We have done that already. So this is structure of an ATP molecule. Now let's see. Contains nitrogen, yes or no? DNA nucleotides with adenine, obviously this is a nitrogen base, so nitrogen is present. RNA nucleotide with adenine, obviously the nitrogen will be present. ATP, obviously it has adenine nitrogen base, so this will have nitrogen present in it. Contains a pyrimidine base. What are the pyrimidine bases? Pyrimidine bases are cytosine, thymine, and uracil. So DNA nucleotides with adenine, obviously adenine is not a pyrimidine, so no. RNA nucleotides with adenine, so adenine is obviously not a pyrimidine, it is a purine, so no. Or ATP contains a pyrimidine base. ATP, because it has an adenine, so obviously no. It is, purine base. Number of phosphate groups. DNA nucleotide with it has 1. RNA nucleotide it has 1, and ATP it has 3. Name of the sugar component. DNA main it is deoxyribose, or RNA main it is ribose, or ATP main it is ribose as well. Okay, so this is the comparison in between these 3 nucleotides: RNA nucleotide, DNA nucleotide with adenine, and ATP. So this is again a comparison in between these nucleotides. Nucleotides are structural component of nucleic acid. Each nucleotide consists of a pentose sugar, a phosphate group, and a nitrogenous organic base. Complete table 5.1 to compare DNA nucleotides with the RNA nucleotide as structural component of nucleic acid. So pentose sugar present DNA में होगी pentose sugar, और उसका नाम क्या होगा? That is deoxyribose. और RNA में कौन सी होगी? That would be ribose. Purine base DNA में कौन सी होंगी? Purine bases कौन सी हैं? It is cytosine, it is thymine, and it is uracil. Sorry, this is purine base. So purine bases are adenine and guanine, and this is pyrimidine. So this is cytosine, thymine, and uracil. Okay, so DNA and RNA have the same purine bases, so there is no difference in purine bases. So both have adenine and guanine, but there is difference in pyrimidine bases. Cytosine is same in both, but DNA has thymine and RNA nucleotide have uracil. So that's a comparison in between DNA nucleotides and RNA nucleotides. Now look at this nucleotide. Figure 3.1 is a diagram of a monomer of a nucleic acid. It is messenger RNA. So this is an RNA actually. Name D. The question is beginning with name D, E, and F. It is not type. So pentose is a type. Pentose is not a name, it is type. Name is what? Name is ribose or name is deoxyribose. So as it is an RNA, so E should be ribose, or F should be a phosphate group. Okay. And D is a nitrogen base, but nitrogen base obviously is a type, and it is a single ring, so it should be a pyrimidine base. और pyrimidine में आपको पता है कि RNA में यूरासिल हो सकता है या फिर साइटोसिन हो सकती है. क्योंकि उसने नाम पूछा है यहाँ पे, तो you should write it, you know. वैसे तो जाहिर है अगर आप इसे pyrimidine लिखते हैं क्योंकि ये सिंगल रिंग है. ओके, ये अगर आप pyrimidine लिखते हो तो ये सिंगल रिंग है, बट दिस इज टाइप क्लास. फिर pyrimidine के अंदर जो है वो और नाइट्रोजन बेस की दो टाइप्स है ना. तो नाइट्रोजन बेस जो है वो भी जाहिर है क्लास है, और क्लास ऑफ मॉलिक्यूल्स नाइट्रोजन बेसिस. फिर pyrimidine के आगे जो है तीन टाइप्स हैं, साइटोसिन, थायमीन एंड यूरासिल. थायमीन विल नॉट बी प्रेजेंट इन आरएनए, नॉट इन आरएनए. इन दोनों में से कोई चीज जो है वो आरएनए में हो सकती है. सो वो आप कुछ भी लिख लें, साइटोसिन लिख लें या यूरासिल लिख लें, सो दैट डजंट मैटर. ओके. During the mitotic cell cycle, free nucleotides are used for the synthesis of both types of nucleic acid, RNA and DNA. Complete sentence A, B, C, D to provide information about nucleotides and the synthesis of nucleic acid. Write the correct terms in the space provided in each sentence. Each nucleotide has three main components. कौन-कौन से main components होंगे? Number one, a group. Number two, 5 carbon sugar. So this 5 carbon sugar obviously should be pentose and a nitrogenous base. So this is phosphate group. The nitrogenous organic base of a nucleotide is either a purine or a, so obviously the other one is pyrimidine. In a DNA nucleotide, the sugar is deoxyribose, and in RNA nucleotide, the sugar is ribose. And the synthesis of RNA from a template strand of DNA is known as, so this we'll discuss in the next lesson, but this is known as transcription. Synthesis of an mRNA. We'll discuss this transcription and okay. So figure 5.2 is an incomplete diagram of an ATP molecule. The base shown in figure 5.2 has a double ring structure. State the term for a base that has a double ring structure. So obviously this is purine base. Single ring hogi to pyrimidine, or double ring hogi to purine. So this is the purine base. और चूंकि ये ATP molecule है, तो this will be an adenine, or this will be ribose. Okay, so once you have understood the structure of nucleotides, now let's see structure of a DNA molecule. Structure of a DNA molecule. A DNA molecule has many nucleotides joined together, which make a polynucleotide chain. And these polynucleotide chains are held anti-parallel to each other, and both these strands are coiled around its axis. So if I draw a DNA molecule, that would appear like this. So this is known as a double stranded helix. Okay, so a DNA molecule, this is a DNA molecule. A DNA molecule is a double-stranded helix. In this DNA molecule, the two strands are held anti-parallel to each other. We look at what does this anti-parallel mean. So you see, if this is one strand and this is another strand. So if this strand is facing, we'll discuss what does this mean. Other is this. So two strands are anti-parallel to each other. और ये जो strands हैं, these strands are made up of nucleotides. So these are also known as polynucleotide strands. So if we look at these strands are held together by hydrogen bonding in between them. So if this is an adenine here, if this is guanine here, if this is cytosine here, this will have thiamine here, this will have cytosine here, and this will have guanine here. So there is hydrogen bonding in between these. Okay. So the two strands are held together by hydrogen bonding. And this hydrogen bonding is specific. ये specific है between complementary strands और वो complementary bases और वो क्या है कि A will always make double hydrogen bond with thiamine, and guanine will always make triple bond with cytosine. Okay, so this is complementary base pairing. Complementary base pairs. And this is triple hydrogen bonding. And this is double hydrogen bonding. Now, what does this anti-parallel mean? इसमें ये जो DNA molecule है, इसमें anti-parallel का क्या मतलब है? Anti-parallel. If I draw a nucleotide again, so let's have a look at, the structure of a nucleotide again before we discuss this anti-parallel. So this is, for example, a pentose sugar. This pentose sugar has an oxygen here, and this is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, and this is carbon number 5 here. Okay, and the phosphate group is attached here with this. And this has an OH group here. The rest we don't need to, as there is no. So this is one nucleotide. This will have, a nitrogen base here. कोई भी nitrogen base हो सकती है, guanine हो सकती है, कोई भी हो सकती है. So this is a nucleotide. Now, if there is another nucleotide, such a nucleotide, and there has to be bonding that takes place in between these. Okay, look at this. If the bonding has to take place in between them, the condensation will take place in between these two molecules here. Okay. A hydroxyl group from one and hydrogen from the other will be removed, and a bond will be formed in between these two. So this bond formed in between these two. Okay. So do you see there is a phosphate group, phosphorus in between, and there is O binding with this carbon number 5 on this end and carbon number 3 on this end. So this bond is known as phosphodiester bond or linkage. Got it? This is phosphodiester linkage. Now, if you look at, if another has to be attached with it, obviously, if there is another attached here, that will also have, you know, this will have so this is another phosphodiester linkage. So this would make a long chain. Okay, so this long chain can be represented as this is sugar, this is phosphate group, this is sugar, this is phosphate group, this is sugar, this is phosphate group, and this is sugar, and this is phosphate group, and this will have a nitrogen base here, which may be, you know, adenine, this may have guanine, this may have cytosine, this may have thymine. Okay, so this is a sugar phosphate backbone. In this sugar phosphate backbone, you know, this is a sugar phosphate backbone. Do you see the phosphate group is towards this 5 side, and this OH is, this OH on the lower side is carbon number 3. So upward is carbon number 5. So this is known as 5 terminal, and this is known as 3 terminal. So maybe this is 5 prime and this is 3 prime. अगर हम represent करते हैं तो इसको इस तरह से हम 5 to 3 direction में represent करेंगे. So this may be 5 and 3, and the other will be 5 on this end and 3 on this end. So this is 5 prime and 3 prime. So that is how these two strands are anti-parallel to each other. Now, if this has to be made, you know, opposite to this, you know, ये अगर इस तरह से है तो इसके against अगर आपने दूसरा strand बनाना हो तो that would be anti-parallel to it. Anti-parallel means, let me just take a copy of it. So we will have so do you see this is how these strands are complementary to each other. This has 5 terminal up, and this is a 5 terminal low below, and this has 3 terminal here, and this has, you know, 3 terminal here.
Sir, how did we decide कि 5 terminal क्या है और 3 terminal क्या है?
I said we have this pentose sugar. This pentose sugar has carbon number 1 here. You know, as we discussed the structure of glucose, so this is carbon number 1 here, this is carbon number 2 here, this is carbon number 3 here, this is carbon number 4 here, this is carbon number 5 here. So every pentose sugar has 5 up and 3 down. So this chain has, you know,
Okay. Okay, got it.
yeah. So that's what makes this. So this is how these two strands are anti-parallel to each other. So there would be double hydrogen bonding here. This would be triple hydrogen bonding here. This would be triple hydrogen bonding here. And this would be double hydrogen bonding here. So this is how the two strands are held anti-parallel to each other in a DNA molecule that later coil around their central axis, making a double helix. Hope you have understood it. So let's see, we have this structure here now. Figure 5.1 shows part of a DNA molecule. Name U to X. Can you identify these structures? U क्या है, W क्या है, और X क्या है?
U is phosphate group.
Yes, this is phosphate group. And W is?
The nucleotide.
No. How nucleotide? Nucleotide will be this whole structure that has been already. No, this is one nucleotide.
Deoxyribose sugar.
This is deoxy, because this is DNA, so this is deoxyribose. You should not write pentose. And what is an X?
Cytosine.
Yeah, because this is guanine already there. So the complementary base will be cytosine. So this should be cytosine here. And name the bonds indicated by Z. So there are bonds here. These are hydrogen bonds. And what are these bonds here? ये जो bonds हैं यहाँ से यहाँ तक, ये क्या है? ये phosphodiester bonds हैं. ओके, तो इसमें देखें, ये जो शुगर है, this is phosphate downwards and this is phosphate upwards. So this is, you know, if I draw this, so this would be, you know, this would be ये जो डायरेक्शन होगी ये इस तरह से होगी. 5 इस तरफ है और 3 इस तरफ है, और इसका 5 इस तरफ है और 3 इस तरफ है. You got it?
Yes, sir.
Now let's see. Figure 1.1 is a diagram showing the structure of a section of a DNA molecule. Draw a circle around one monomer of DNA. तो एक मोनोमर जो है, एक न्यूक्लियोटाइड उसके गिर्द जो वो सर्कल आप ड्रॉ करें, तो वो कोई भी आप ड्रॉ कर लें, यहाँ से लेके ये पूरा, ये एक. Name the two bases forming the base pair at X. X पे जो base pair बन रहा है ये वाला. Name the two base pairs forming the base pair at X and give the reason for your answer. तो ये क्या होंगी ये दो?
This is going to be G and C.
Yes. Why?
Because triple bond.
Yes, there is a triple hydrogen bond. So it is GC pair. Or you can write guanine and cytosine. Describe the structure of DNA. जो अभी हमने डिस्कस किया है वो सारा अब हमने अगर डिस्क्राइब करने के लिए आ जाए तो क्या कहेंगे? हम ये कहेंगे कि एक तो डबल हीलिक्स है और स्ट्रैंड्स आर हेल्ड टुगेदर बाय हाइड्रोजन बॉन्ड्स, और हाइड्रोजन बॉन्ड जो वो कॉम्प्लीमेंट्री बेस पेयरिंग के बीच में होगी, एडेनिन और थायमीन में और साइटोसिन और गुआनिन में. और ये जो स्ट्रैंड्स हैं वो एंटी पैरेलल होंगे. ओके, और ई स्ट्रैंड एज अ शुगर फॉस्फेट बैकबोन जो फॉस्फेडाइस्टर बॉन्ड्स होंगे इसके दरमियान में, और मोनोमर यूनिट्स डीएनए आर डीएनए न्यूक्लियोटाइड्स और पॉलीन्यूक्लियोटाइड स्ट्रैंड, और न्यूक्लियोटाइड जो है वो होगा डीऑक्सीराइबोज शुगर, फॉस्फेट ग्रुप एंड एनी ऑफ द नाइट्रोजन बेसिस. और पॉइंट्स ऑन द ड्राइंग diagram, you can make a diagram and you can describe on a diagram as well. जो ऊपर हमने diagrams देखी हैं वैसे आप diagram भी draw कर सकते हैं, या फिर आप ये describe कर दें ये points. So that's all about it for today. अब ये DNA double कैसे होता है, DNA duplication कैसे होती है, that we'll discuss in the next class.
Sir, can you please share this document in the group? Like the PDF for the questions.
Okay. Solved or unsolved?
Sir, I mean whatever you have, अगर solved है तो ठीक है, नहीं तो unsolved भी.
नहीं, दोनों ही है मेरे पास.
Sir, फिर solved ही दे दें.
सॉल्व तो रिकॉर्डिंग में आ जाएगा ना आपके पास. मैं रिकॉर्डिंग शेयर कर देता हूँ, और अभी आपको मैंने भेज दी है अनसॉल्व. इसको आप खुद से सॉल्व करें.
Okay, sir.
ठीक है, ये मैंने भेज दिया आपको. सो दैट्स इट फॉर नाउ. आई एम एंडिंग दिस सेशन. अल्लाह हाफिज.
Love this.